For centuries, the letter O has appeared on medical forms as if it were nothing more than a routine detail. A doctor sees it, a laboratory records it, and a patient moves on. But behind that single letter is a genetic story connected to mutation, migration, disease, and the deep history of our species. And if you are black and have type O blood, there is something important you should know before believing the viral claim that O is simply the oldest blood type. That claim is too simple. Type O does not automatically reveal a person's ancestry, and it does not prove that one specific African mutation created every O blood type found around the world. Modern genetics shows that several O variants exist and that blood group evolution is more complicated than the popular story suggests. But that correction does not make the story less fascinating. It makes it more fascinating. Because when scientists stop looking at the letter O alone and examine the wider DNA surrounding African populations, they uncover something far more powerful. Africa contains the greatest overall genetic diversity of modern humans, some of the deepest maternal lineages ever identified, extraordinarily old paternal branches, and genetic adaptations shaped by diseases that affected African populations for thousands of years. In other words, your blood type may not be a secret ancestry code, but it sits inside a much bigger biological history that reaches directly into the origins of our species. And that story does not begin in Europe, Asia, or the Americas. It begins in Africa. The ABO blood group system was discovered in the early 20th century, when Karl Landsteiner observed that blood from different people did not always behave the same way when mixed. Some combinations caused red blood cells to clump together while others did not. That discovery eventually produced the familiar categories A, B, AB, and O. We now know that the system is controlled by the ABO gene on chromosome 9. The gene produces an enzyme that modifies sugar molecules on the surface of red blood cells. In type A, one form of the enzyme adds the A antigen. In type B, another form adds the B antigen. People with both forms have AB blood. In the common O form, the enzyme is disrupted, so the A or B modification does not occur, leaving the red cells without A or B antigens. One famous O allele contains a small deletion that changes the reading frame of the gene and destroys the enzyme's normal function. But this is where the popular story needs correction. There is not one universal O mutation that appeared once in Africa and then traveled unchanged into every O-positive person alive today. Several O variants exist, and they have different evolutionary histories. Some are common in particular regions, others are rare, so the letter O cannot identify one ancient population. It cannot tell you whether your ancestors were Igbo, Yoruba, Wolof, Akan, Congo, Mende, or another African population. It cannot tell you which country your ancestors came from. What it can do is give us one small clue inside a much larger story about human genetic variation. The real surprise begins when we zoom out from the ABO gene and ask a much bigger question. Where is the greatest human genetic diversity found? The answer is Africa. That conclusion has been replicated across many forms of genetic research. Africa contains the deepest overall diversity because Homo sapiens has been evolving there for far longer than populations outside the continent. When relatively small groups of humans left Africa and founded populations elsewhere, they carried only part of the variation that existed in the larger African population. Geneticists call this a founder effect. Imagine a library containing 10,000 books. If a small group leaves carrying only 500, the new library can grow and collect new books, but it will not begin with all 10,000. The original library contains much more of the historical collection. Human populations followed a similar pattern. The people who moved out of Africa experienced bottlenecks, and those bottlenecks reduced some of the genetic diversity their descendants carried. This is why populations outside Africa generally contain less genetic diversity than the African continent as a whole. That does not mean Africans are more advanced, more intelligent, or biologically superior. Genetic diversity is not a measure of human worth. It is a record of demographic history. It tells us where populations lived, how large they were, how often they mixed, and how much variation survived from one generation to the next. When you understand that, the African genetic story becomes extraordinary. The deepest maternal branches provide one of the clearest examples. Mitochondrial DNA is passed from mothers to their children, allowing scientists to trace direct maternal lineages. The oldest branches of the human mitochondrial family tree are African. Haplogroup L2 contains some of the deepest known maternal lineages, especially among populations in southern Africa. Certain Khoisan communities carry particularly ancient L0 branches. Scientists have estimated that some of these lineages diverged from other human maternal branches well over 100,000 years ago, although exact dates vary depending on the mutation rate and statistical model being used. That does not mean that today's Khoisan are unchanged descendants of people who lived 100,000 years ago. They are modern humans. Their languages, societies, environments, and genes have continued to change. The importance of those lineages is that they preserve an exceptionally deep branch of the human maternal tree. Every maternal lineage found among people living outside Africa ultimately fits inside the much broader African mitochondrial history. That is a powerful scientific fact, but it is essential not to turn it into a racial hierarchy. The meaning is not that one group is older as people. Every living human is equally modern. What is older is the population branch. Some branches split earlier than others. Africa contains many of those early branches because human evolution happened there for such a long time. The paternal side of the story provides another dramatic example. The Y-chromosome passes from father to son, allowing scientists to follow particular male lineages. In 2013, researchers investigating an unusual Y-chromosome found in an African American genealogy participant identified an extremely rare branch called Azorojero. It was so deeply separated from known Y-chromosome lineages that the researchers initially estimated its divergence at roughly 338,000 years ago. Later studies refined the picture and showed that these dates depend heavily on assumptions about mutation rates and population history. But the central discovery remained extraordinary. Azorojero was an early branching paternal lineage that had previously escaped the global genetic record because so few carriers had been sampled. Researchers later found additional Azorojero carriers among people with ancestry from western Cameroon. The discovery was important not because it proved that every African man descends from AR0, but because it demonstrated how much of the human family tree remained hidden inside undersampled African populations. A lineage could exist for generations without being represented in a major database. Then one unusual DNA sample could reveal a branch deeper than scientists expected. This is one reason modern African genomics is so important. Every additional population sampled has the potential to reveal new variants, new branches, and new demographic histories. The genetic map of humanity is still incomplete. Now consider another blood-related trait that tells an even stronger story about African environmental adaptation, the Duffy blood group. The Duffy antigen is controlled by the ACKR1 gene and is expressed on red blood cells. A common regulatory variant, often called the Duffy-null allele, greatly reduces or eliminates Duffy antigen expression on red blood cells. This phenotype is extremely common in many sub-Saharan African populations, but much less common elsewhere. Why would that happen? One explanation involves malaria, especially Plasmodium vivax, a parasite that uses the Duffy receptor during invasion of red blood cells. If red blood cells lack that receptor, the parasite's traditional route of entry becomes much harder. This created a strong selective advantage in regions where the parasite was common. Over many generations, the Duffy-null variant became very frequent in parts of Africa. Modern research has added an important qualification. Duffy negativity does not guarantee complete protection from PVIVAX because infections can occur through alternative mechanisms. Still, the overall evolutionary relationship remains clear. A human population faced a repeated disease threat, and a genetic variant that reduced susceptibility became extremely common. That is natural selection written into blood. It shows how environment can leave measurable fingerprints in the genome. The blood of African populations contains not only ancestry from ancient people, but also adaptations created by the diseases those people survived. Sickle-cell trait tells the same story from another direction. A variant in the HBB gene can cause sickle cell disease when inherited in 2 copies. But carrying 1 copy can provide protection against severe malaria. This creates a classic evolutionary trade-off. In areas where malaria is widespread, individuals with 1 copy can have better survival than people without the variant. Natural selection therefore maintains the allele in the population. Yet when 2 copies are inherited, the medical consequences can be severe. This is why the variant did not simply spread until everyone carried it. Evolution does not pursue perfect outcomes. It balances survival advantages against biological costs. The sickle cell story is especially important for people of African descent because the variant is common in regions of West and Central Africa where malaria historically exerted strong evolutionary pressure. But again, it is wrong to treat sickle cell trait as a racial identifier. Similar variants occur in other populations exposed to malaria, including parts of the Mediterranean, Middle East, and South Asia. The relevant factor is ancestral exposure to malaria, not skin color. This distinction matters because modern medicine should rely on direct genetic testing rather than racial assumptions. Ancestry can change probabilities, but it cannot tell us exactly what is inside one person's genome. APOL 1 gives us another example of ancient survival with modern consequences. Certain APOL 1 variants became common in parts of sub-Saharan Africa because they improved resistance to parasites responsible for sleeping sickness. But inheriting certain combinations can also raise the risk of kidney disease. Once again, evolution created a trade-off. A mutation that helped ancestors survive an infectious threat could contribute to medical problems in a different environment. This pattern is not unique to Africa, but Africa contains many striking examples because of the intensity and diversity of infectious disease pressures across the continent. Malaria, trypanosomes, and numerous bacterial and viral infections have shaped human populations for thousands of years. The result is a genome rich in immune and metabolic variation. Some of these variants may protect against disease. Others may become liabilities in modern environments. That is why African genetic diversity is medically important. Researchers who study African populations continue to discover variants that are rare or missing from older European-dominated databases. The more African genomes are sequenced, the more accurate our understanding of global human biology becomes. This brings us back to type O. Several studies have investigated whether ABO blood groups influence malaria severity, and there is evidence that type O is associated in some settings with a lower risk of severe malaria compared with certain other blood groups. One proposed mechanism involves the way infected red blood cells interact with other blood cells in the bloodstream. But this relationship is not absolute. Type O does not protect a person from malaria, and the effect varies by parasite, region, age, immune status, and other genetic factors. This is another example of why the popular claim that blood type reveals your ancestral destiny is too simplistic. Blood groups are influenced by evolution, but they are only one part of a much larger network of genes. The human body contains thousands of variants that interact with each other. A person with O blood might also carry sickle-cell trait, Duffy-null, APOL1 variants, or none of them. Another person with A blood might carry entirely different African adaptations. The most useful information comes from the complete genetic profile, not a single letter. The real wonder is that these blood-related systems can all preserve evidence of different chapters in human history. One gene reflects ancient mutation, another reflects malaria, another reflects migration, another reflects population growth. Your body is not just functioning, it is also carrying the accumulated consequences of what your ancestors experienced. For people of African descent outside Africa, this becomes especially meaningful because so much of the historical record was deliberately damaged. The transatlantic slave trade forcibly removed millions of Africans from their homelands and carried them across the Atlantic. Those people came from many different parts of West and Central Africa. They were not one ethnic group. They spoke different languages, belonged to different political systems, practiced different religions, and came from societies with their own histories. Yet the slave system often reduced them to numbers, commodities, and short descriptions. Family connections were destroyed, names were changed, written records were inconsistent, entire ancestral identities disappeared from the documentary trail. But DNA was carried across the ocean. The people who survived the Middle Passage still carried their maternal lineages, paternal lineages, immune variants, blood group alleles, and countless other genetic signatures from Africa. Their descendants inherited those signatures even when they no longer knew the names of the communities that produced them. Modern genetic research cannot recover every lost ancestor, but it can recover population signals. It can sometimes show that a person has ancestry associated with West Central Africa, the Bight of Biafra, the Gold Coast, Senegambia, or other regions. It can reconnect living people with broad geographic histories that written records had obscured. The genome becomes a second archive, one that survived even when the paper archive failed. That is why genetic ancestry testing can be so powerful for descendants of the African diaspora. A DNA result does not replace genealogy, but it can open doors. Genealogy cannot. If historical documents stop at an enslaved ancestor with no clear birthplace, genetic evidence may identify broad ancestral regions. If a family has oral traditions about a particular area, DNA can sometimes support or complicate those traditions. If distant relatives share long genetic segments, researchers can identify family connections that paper records never documented. The process is not perfect. Commercial ancestry tests rely on modern reference populations, and historical African populations do not always match modern borders or ethnic groups. A percentage associated with Nigeria does not automatically mean one ancestor was Igbo, just as a result associated with Ghana does not prove one ancestor belonged to one specific Akan group. Modern ethnic labels are not identical to ancient populations, but the fact remains that genetic information can restore part of the map. It can show that the ancestral story is not blank. It is simply complex. The African genetic story becomes even more powerful when viewed through the global family tree. Humans outside Africa are descendants of relatively small groups that left the continent during ancient migrations. The exact timing and structure of those migrations remain debated, and there were likely multiple dispersals, including some early groups that contributed little to later populations. But the main ancestry of today's non-Africans still traces back to African populations. Founder effects reduced genetic diversity as people moved into new regions. Later, populations encountered Neanderthals and Denisovans and exchanged genes. They adapted to new climates. They developed new diets, immune systems, skin pigmentation patterns, and metabolic strategies. But the underlying history still points back to Africa. That means the African genome is not a side branch of human evolution. It contains the largest reservoir of human variation. When scientists sequence a previously underrepresented African population, they often find variants never seen in global databases. These discoveries can change the shape of the human genetic tree. They can also reveal ancient population structure that disappeared elsewhere. This is why African ancestry is scientifically important, far beyond questions of identity. It is central to understanding what humans are. If you are black and have type O blood, therefore, the scientifically accurate message is not that O makes you more ancient than everyone else. It is that your blood group is one piece of a much larger genetic history. Your ABO allele has an evolutionary past, but that past is not a personal ancestry certificate. Your mitochondrial DNA may trace a maternal line deep into Africa. Your Y-chromosome, if you are male, may preserve a paternal branch that separated from other lineages extremely far back in time. Your immune genes may carry variants shaped by malaria or other African pathogens. Your genome may contain ancestry from several African regions because the transatlantic slave trade and later migrations combined populations that had once been separated by thousands of kilometers. The real story is therefore bigger than type O. It is about what happened to African ancestry over deep time. Your ancestors did not simply survive. They adapted, they moved, they mixed, they founded new communities, and they passed their DNA forward. There is also an important distinction between ancestry and identity. DNA can tell you about biological relationships, but it cannot tell you what culture you belong to. A person can have substantial African ancestry without identifying with one particular African community. Another person can identify strongly with an African cultural tradition while having ancestry from several regions. Genetics should never be used to police identity. The same principle applies to blood type. O does not may. The same principle applies to blood type. O does not make someone more African. A does not make someone more European. B does not make someone Asian. Blood groups are distributed across populations and shaped by multiple evolutionary processes. If anything, the global distribution of ABO types is another demonstration that human populations have always been connected. Variants move through migration and admixture. Populations change. There are no genetic borders around races. The genome is a continuum of relationships. And there is something else your blood carries that most people never think about: the memory of environment. Imagine an ancestor living in an area where malaria kills children at frightening rates. A variant that slightly improves survival becomes more common. Repeat that process for hundreds of generations, and the variant can become a defining feature of the population. Now imagine another ancestor moving into a new climate, changing diet, encountering new pathogens, and marrying someone from another population. Different variants are introduced. Some disappear, others remain. Eventually, their descendants carry a complex combination of adaptations. This is how human evolution actually works. It is not a straight line. It is a constant negotiation between genes, environment, migration, chance, and social behavior. African populations provide some of the best evidence for that process because the continent contains such deep diversity and such a wide range of environments. From tropical forests to deserts, highlands, savannas, and coasts, African populations faced dramatically different ecological challenges. Their genomes preserve some of the responses. One of the most fascinating consequences of this research is that modern medicine is finally recognizing how much was missed by studying mostly European populations. For decades, large genetic studies were disproportionately based on people of European ancestry. That created reference data sets that were useful, but incomplete. Some disease-associated variants found frequently in African populations were overlooked because they were rare in European samples. Some drug metabolism variants were missed. Some immune variants were underrepresented. As more African genomes enter research databases, the picture becomes clearer. Scientists can identify genetic variants that may influence kidney disease, malaria susceptibility, cardiovascular risk, immune response, and drug response. This does not mean there is one African medicine and one European medicine. It means the human genome is diverse enough that medical models need to reflect that diversity. The more populations scientists include, the more precisely they can predict biological variation. African genetic diversity is therefore not only a story about the past, it is part of the future of medicine. The deeper scientific lesson is that blood is not an ancestry test, but blood can be a window into evolution. The ABO system reveals how one gene can create different biological phenotypes. The Duffy system reveals natural selection against a parasite. Sickle-cell variation reveals a trade-off created by malaria. APOL1 reveals how protection against one pathogen can increase disease risk in another context. Mitochondrial DNA reveals the structure of maternal ancestry. Y-chromosomes reveal paternal ancestry. Whole genomes reveal the broader network connecting all of them. No single marker is the whole story. The human body is a collection of historical records, each written in a different genetic language. When scientists combine those languages, the picture becomes astonishingly clear. That is why the letter O should not be treated as proof of an original bloodline. The better conclusion is much stronger scientifically. If you have African ancestry and type O blood, you carry one of the world's most widespread blood group phenotypes inside a genome connected to one of the deepest reservoirs of human genetic diversity. Your blood may contain genetic variants shaped by diseases that challenged your ancestors. Your chromosomes may contain ancestry from multiple African populations. Your maternal line may trace through one branch of the ancient African mitochondrial tree. Your paternal line, if you are male, may trace through a different ancient branch. And if your ancestors were part of the African diaspora, your genome may also preserve a record of the catastrophic migrations that reshaped the modern world. That is a much bigger story than one letter. It is a story of origin, a story of adaptation, a story of migration, a story of survival, and a story of memory. Your ancestors did not leave their history behind when they moved. They carried it. They carried it through forests, across deserts, across rivers, across borders that did not yet exist. And for millions of people, they carried it across an ocean through one of the darkest chapters in human history. The names were lost, the languages were suppressed, the records were broken. But the DNA continued. A mutation survived, a maternal lineage survived, a paternal lineage survived, an immune adaptation survived, a blood group allele survived. Generation after generation, the biological record continued forward. And now, scientists can read parts of it. That does not mean your blood type tells you everything about where you came from. It does not. But it does mean that the genetic story inside your body is much older than your surname, older than your country, and older than almost every institution you know. The deepest branches of the human genetic family tree are African. The greatest overall human genetic diversity is African. Some of the oldest maternal and paternal lineages still exist in African populations. Many important disease adaptations were shaped by African environments, and the descendants of those populations now live across every continent. So the next time you see the letter O on a medical chart, do not imagine it is an ancient passport. Think of it as one small symbol in a much larger genetic archive. The real ancestry story is written across thousands of genes, It is written in the mutations you inherited. It is written in the adaptations your ancestors survived. It is written in the populations they joined. And it is written in the descendants who came after them. Your blood does not tell the whole story, but it is carrying part of it. And that part began long before any modern country existed, long before any modern racial category existed, long before the first written history in Africa. And the more scientists learn to read the genome, the clearer that ancient beginning becomes.